Hi, so we're back for photosynthesis. Now, there we go, it's all working. So, what's the purpose of photosynthesis? Well, amazingly, of course, without photosynthesis, there is no life on this planet. The purpose, of course, is to capture light energy, convert it into stored chemical energy, so that we can then break it down and release it in a form through, say, respiration that we can make use of. So, without this little diagram here, I'll get myself out of the way for a minute. I'll put myself in the top corner there. With that little diagram here, this chemical reaction, there'd be no life on this planet. 6 carbon dioxide molecules mixed with 12 water molecules in the presence of light and chlorophyll, otherwise we'd be raining sugar in the classroom, produces glucose, there's my thing up, glucose, 6 water, and 6 oxygen. And these guys, of course, are critically important because when plants started doing this, we changed our atmosphere considerably, and life became much more pleasant for living things on the planet. So we're using up CO2 and creating O2. And, of course, most importantly, chemical stored energy. So critically important. It occurs in things called chloroplasts, and chloroplasts are simply one of our organelles, well, found in plants, not in mammals. And because it has its own DNA, we believe it actually was a prokaryotic cell which is taken up by later evolving eukaryotic cells. So we call this living in symbiosis. It was stayed symbiosis last year, and this is what we call endosymbiotic theory. So inside, not outside. This is an organelle within another cell. So what's the evidence? Well, there's 3 main points for this theory. These chromosomes, these chloroplasts here, have a double biphospholipid layer, like all cells. They also have their own free-floating DNA, just like a bacterial cell. The prokaryotic cells, bacterial cells, have free-floating DNA, and therefore they're able to self-replicate. And so cells in plants that have a lot of light exposure will have lots of chloroplasts. Cells without any light exposure don't have them at all, and cells with low light have less. And they should respond to the environment in that way. So photosynthesis occurs in essentially 2 stages. It's a light-dependent stage and a light-independent stage. So the chlorophyll traps the light energy, and it uses it to produce ATP and to split the water molecules. So we get 24 hydrogen ions and 6 oxygen gas molecules. So the oxygen gas leaves in this first stage. That waste is created straight away. And this part occurs in what's known as the stroma of the chloroplast. Those 24 hydrogen ions are then combined in the light-independent stage with the 6 carbon dioxide ions, molecules, to form our glucose molecule and the waste water. You'll see here that, in fact, the outcome is a thing called G3P rather than glucose. Now, in your exam, we're going to be looking for glucose, but G3P is actually the thing that we build all of the glucoses and sucroses and things from. So it's actually not glucose at all that's produced. That's this next stage, and it's all driven by rubisco. We've got rubisco on this diagram. They haven't, unfortunately. There's an enzyme called rubisco which drives this whole process of light-independent stage of the cell, of the plant cell. And of course it has some limiting factors. You need light. Yeah, I know. It's pretty obvious, isn't it? No light and photosynthesis won't occur. Some plants living in really dry conditions will do the light-independent stage, so do the light-dependent stage when the light's around and do the light-independent stage later, so as to control water loss. Clearly, if we have low light, then the rate of photosynthesis is lower. If this is boundless light, then as long as there's CO2 and water present, you get boundless production of glucose. That sort of makes sense, doesn't it? Water is also a critical ingredient, and so low water levels causes problems for plants. If there's low water, then they tend to close down the stomata. If you close your stomata to control water loss, you stop gas exchange and you stop photosynthesis. However, if water is plentiful, then you can leave your stomata wide open gases exchange freely and you can photosynthesize to your heart's content. Although, of course, they're plants, they haven't got hearts. Shouldn't have said that. The other limiting factor clearly is carbon dioxide, because you're building your glucose from carbon dioxide and water with the presence of light. So if carbon dioxide levels are low, clearly photosynthesis will slow down. If concentrations of carbon dioxide are high, then clearly you get lots of glucose being produced. And interestingly enough, some of the early plants have a form of rubisco, the enzyme that controls all this process, that are supercharged. They actually can convert more CO2 from the atmosphere than the rubisco we see in most highly evolved plants that now live on the environment, in the environment. And there is some work being done by geneticists to try and include supercharged rubisco in our food crops. Having been trouble with the architecture of modern plants, doesn't quite carry the supercharged rubisco gene correctly, but they're working towards trying to find crops which will take that supercharged rubisco and take more CO2 out of the atmosphere and create good food crops. So resolving climate change to some degree, and also helping resolve food availability for people as well. And that's the end of photosynthesis, another nice little short one.